All right, I think we might kick off. Thanks everyone for joining our first spatial conversation webinar for the year. I'm Brett Kennedy. I'm the Market Development Manager for ANZ for Spatial from 10x Genomics, and I'm joined as a host by Marta Gabrielski from Millennium Science. For those of you who are new to this format, the idea is really to get more of the background information, unpublished details about performing spatial experiments into the public domain. It's a fast-moving field. We know it can be sometimes hard to get information. So, we hope that you can find these talks valuable. And we encourage you to ask as many questions of our speakers as possible, as they've put a lot of time and thought into these experiments, and we're sure they have a lot to share that you normally can't access outside of one-on-one conversation. Marta.
Yes, welcome everyone. So thank you, Brett, for introduction. Millennium Science is distributor of 10x Genomics, among other suppliers, but 10x is my favorite, obviously. And I would like to just say that if you have questions during our webinar, please put them in the chat, preferably so that directed to everyone, so everyone can see and not maybe duplicate the questions. We will answer all of them. And now I would like to introduce our speaker, Jeremy Moe, PhD student and medical oncologist from Garvan Institute. He will be talking about spatial transcriptomics and tumor microenvironment during chemotherapy. I think the topic is super interesting. So Jeremy, please, the floor is yours.
Thank you for the kind introduction, Marta. And thanks everyone for attending. I'll just share my screen so we can get this started. Great. So I've got a bit of a wordy title for today's talk, and hopefully it'll become simpler as we go along. Well, we're speaking about how spatial transcriptomics unveils disparities in the tumor microenvironment and its dynamics during neoadjuvant chemotherapy. So I'm currently a PhD student at the Garvan Institute, being supervised by Alex Swybrick and Elgene Lim. So to start with just a bit of background, the tumor microenvironment is heterogeneous, and we recognize that it's comprised of a multitude of elements, both cellular and acellular components, with particularly cancer-associated fibroblasts, immune cells such as lymphocytes and myeloid cells, endothelial cells, parasites, and adipocytes, as well as acellular components such as the extracellular matrix. And we know that through treatment and time, the tumor microenvironment can change, with therapy inducing heterogeneity in the tumor microenvironment, with an increasing focus on the changes that occur with treatment. And there have been several publications using spatial technologies to study these changes and the remodeling associated with therapy within the tumor microenvironment. So the aim really of our current experiment has been to map tumor microenvironment dynamics in breast cancer across time and treatment. And we're utilizing samples from patients undergoing neoadjuvant systemic therapy for their breast cancer. And what we're hoping to compare is the differences in tumor microenvironment characteristics between responder and non-responder, and I'll explain a bit about what that is in a moment, as well as between different breast cancer subtypes because of differing patterns of treatment resistance. So neoadjuvant systemic therapy refers to the use of treatments such as chemotherapy or targeted treatments prior to localized therapy like surgery. And currently it is considered the standard of care for a subset of early breast cancers, such as HER2 positive or triple negative breast cancer. The utility of this really is that the surgical pathology can be used as a surrogate for long-term patient outcome, particularly if the patient achieves what we refer to as complete pathological response, where there are no viable tumor cells present within the surgical specimen. That does correlate with long-term survival outcomes, such as overall survival and event-free survival, both which are used as clinical measures. And it's been in use or been reported in use since 1997, where we showed that it had outcomes that were similar to the use of adjuvant treatment, which is after localized therapy such as surgery. And subsequent multi-center pooled analyses showed that the surgical outcomes, complete pathological response, or having residual disease, where there are viable tumor cells present, correlating with long-term survival outcomes. So we wanted to use this setting, the neoadjuvant setting, to try and uncover some of these temporal and treatment-induced changes within the tumor microenvironment. And we have, through our patients, a number of samples collected at different time points. And again, these are patients of two clinicians, Professor Elgin Lim and Dr. Mun Huey, both who are medical oncologists. And I'll speak about when these samples were taken. So you can see here that we've got biopsies taken pre-treatment, mid-treatment, as well as also samples taken from time of surgery. And what we refer to as mid-treatment is between two regimens of chemotherapy, the first being doxorubicin and cyclophosphamide, followed by paclitaxel-containing treatment, which would have been with or without carboplatin, depending on the subtype of cancer. And so again, just referring to the three different time points that we've taken our samples. So the samples that we've obtained, aside from the biopsies, we also have a number of other tissue samples, which we've used for single-cell transcriptomics. And through our tissue, our FFPE samples, we've proceeded with performing spatial transcriptomics utilizing Xenium. And in terms of the number of samples that we have 4 patients with triple-negative breast cancer who have achieved a complete pathological response. We have pre and mid-treatment. And for residual disease, we have both triple-negative and luminal B or hormone receptor-positive breast cancer. And we have samples that were taken pre, mid, as well as from time of surgery. And the reason why there isn't a time of surgery time point for those with complete pathological response is because without viable tumor, it's hard to say what is still part of the tumor microenvironment. In terms of the panel design, this again was a big headache during the process to try and think about what markers and genes we wanted to include in our standalone panel. But the way that we approached this was by firstly taking the 10x multi-tissue panel, removing redundant genes or genes that were irrelevant to our study, and then adding in markers that we had already from in-house single-cell references, public single-cell references, as well as adding in genes of interest, including stromal phenotypic markers, as well as stromal pathway markers, such as TGF-beta, and I'll come back to that as well later. The biological questions which we're hoping to answer with this were to, again, evaluate the tumor microenvironment dynamics with treatment, which includes cell composition change, as well as cell-cell communication, and comparing between those two biological settings, comparing between triple-negative responders, those with complete pathological response, and non-responders, those with residual disease, but also comparing between a triple-negative breast cancer non-responder and a luminal B non-responder to see how treatment resistance may differ between those two different subtypes. And so here is an example. I'll start off with looking at the same slide H&E image. So again, this was from a patient with triple-negative breast cancer. This was their post-treatment or surgical sample, and they had residual disease. And the reason why I focused on this area is because for those who are familiar with H&E images, we can see that there is an aggregate of immune cells, which may potentially be a tertiary lymphoid structure. So if we then look at the image from Xenium, this is the Xenium image that comes off the machine. We have used the multimodal segmentation reagent. We can then identify in some coloration how the tissue looks. And if we then add on coloration based on segmentation method, we can see that the majority of our cells are being segmented by interior staining or the cytoplasmic staining with RNA staining. We also have about 20% of cells being segmented by boundary staining or the cell membrane. And we had about 5 to 10% of cells which were being segmented by nuclear expansion, or the method that was used prior to the introduction of the segmentation reagent. And if I zoom in again on that area of interest with that potentially tertiary lymphoid structure, we can see that if I color in some of the transcripts or markers that we had on our gene panel, such as CD8A for CD8 positive T cells, Pink is EpCAM for our epithelial cells, and so is keratin 5 or KRT5. And MS4A1 in orange there as a marker for B cells. We can see that indeed that this looks like a germinal center or a tertiary lymphoid structure. So we can see that there are B cells surrounded by CD8 positive T cells. And interestingly, we can see the mingling of these epithelial cells, which are likely invasive cancer cells, as they are not associated with any ducts, as we'd expect as part of breast cancer tissue structure. If we then add on the clustering and cell coloration by clustering, we can then see that the labels that we've transferred, and I'll talk a bit about our annotation methods, indeed show that these cells are as expected, with the B cells surrounded by the T cells, as well as association with those malignant invasive epithelial cells. Now, I'll touch briefly on annotation, and this will be expanded upon by our computational biologist, Hanyun Zhang. She's been working on this project alongside with me, and she'll also be talking in a second part talk to this, talking more about the computational perspective. The way we've approached annotation is firstly, again, using our in-house single cell reference and transferring labels for cells. But we found that a number of cells weren't being accurately labeled. And we had to then compensate for this by then using latent clustering and using clustering-based correction for those cells that didn't have any labels attached. And we assigned major cell type based on this. We then proceeded to further define cells that weren't able to be accurately annotated using a marker gene ranking-based correction, using key markers for each major cell type. And here we can see on our dot plot the number of markers that we've used to annotate our cells. And what I'd just like to draw your attention to is still the fact that there are a number of unknown cells that didn't have high expression in keeping with any particular cell type. And this has been a point of concern. There are about 16,000 cells out of our total number here that you can see on the right-hand side, which were unknown via this clustering and annotation method. And we are looking at other clustering methods which do take in spatial coordinates, as well as using quality control methods to try and see if we can remove these unknown cells or more accurately annotate them. And what we can do with this data is start to examine the cellular dynamics of interest. When we break down our different compartments of cells, you can see here, we've broken them down into major cell types. We've also, on the left-hand side in blue, have the complete pathological response, and in the column next to it, in the red time points, we have residual disease. So we can try and track the trajectories within each of these major cell compartments to see exactly what is occurring across treatment in time. And we can see that in our samples with residual disease, that the B cells seemingly do decrease quite significantly already at the mid-treatment sample for those with residual disease. We can also see similarly for 3 out of our 4 patients with a residual disease that the myeloid compartment also had a significant decrease at mid-treatment. From this, we can also start looking at some ligand receptor analysis to try and see if we can infer cell-cell communication using spatially constrained methods. And the package that we've used here, and I'll talk about, is the COMOT package, which was looking at ligand receptor pairs inferred from cell chat with distance threshold set to 30 microns. On the right-hand side, we have projected our different annotations, and you can see that in green, we have our cancer epithelial cells. And adjacent to that, we have our cancer stroma, which includes cancer-associated fibroblasts. And the 2 pathways that we've included here are some of the more well-known pathways between cancer cells and stroma. So we can see here on the left, GDF15 to TGF-beta receptor 2. We can see that predominantly we have our cancer cells as senders, with the direction of the arrow being the direction of the interaction, and the thickness of the line inferring the expression of the ligand and receptors. And here on the right, we can see that the predominant receivers are the cancer-associated fibroblasts. Whereas if we compare this interaction to TGF-beta 1, to TGF-beta receptor 1 and TGF-beta receptor 2, we can see that the senders really are more diffused throughout the entire tissue, and so are the receivers. We can then start to quantify these cell type specific interactions to try and see what are the major players within the tumor microenvironment, and again, how they're changing. And on this box plot, we have in red the residual disease, and we have in blue the PCR. And how we've broken this down is at pre-treatment, the baseline sample here on the left, and on the right, we have our mid-treatment samples. And what we can see is that mid-treatment, when we compare this to pre-treatment, the number of B cell interactions have decreased significantly for those with residual disease. Although this may be influenced by the decrease in the number of B cells, again, this was an interesting preliminary finding. The other interesting finding was a decrease in the endothelial reactions as well at mid-treatment for those with residual disease. And both of things are points we'd like to explore further without further analyses. We can also start looking at time point specific interactions to see if there's anything that stands out at early treatment or at mid-treatment. And what we found is that, again, for those with residual disease, what we consider as patients not responding as well as we'd like, we can start identifying that there are some specific interactions. And what we found is this interaction between galectin-9 or LGLS9, the gene, and CD44. And this interaction has been known to potentially be a point of driving differentiation of T cells into Tregs. And that itself has also been associated with treatment resistance with both chemotherapy as well as immunotherapy. So it's quite interesting to see that we're already identifying these interactions with our preliminary analyses. What we can also do with our current annotations is compare them with clinical ground truth or pathologist annotations. And these have been provided to us by our labs pathologist, Professor Sandra O'Toole. And on the left, we have the annotations projected on using our Xenium analysis. And on the right, we have the pathologist annotations. And what we found is that areas of normal epithelial cells and areas of invasive epithelial cells actually correlate quite well when we compare our Xenium analysis and cell annotations with the pathologist annotations, which gives us some confidence about the data that we're looking at. So in summary, so far, we've been able to identify dynamic changes in the immune microenvironment throughout chemotherapy with patterns that differ between responders and non-responders. And when we start to map these cellular interactions in space, we see that there are certain ligand receptor interactions specific to mid-treatment time point in residual disease, indicating potential paracrine signaling related to chemotherapy resistance. Where we're trying to head to from here is to utilize QC methods to filter low-quality cells, to trial clustering methods that consider spatial coordinates such as Banksy, to annotate minor cell types, and to integrate our findings with single-cell transcriptomics, and also to validate our cell-cell interactions with not just one method, but with multiple methods to see if they still remain. And these are just my acknowledgements, firstly to patients and carers for provision of samples and participation in clinical trial, as well as my lab, particularly Han Yoon, who will also be speaking potentially in a few weeks about the computational analysis of this data.
Thank you, Jeremy. That was really, really interesting. Just a reminder to everyone in our audience, feel free to pop your questions in the chat. This is spatial conversations, so we need to actually discuss things. And as Jeremy mentioned, we are organizing a follow-up spatial conversations regarding this data focused on bioinformatic analysis. And I'm sure Brett has some questions. I jotted down a few myself. So maybe I will start. I'm just taking over here. So actually, it might be related to the actually bioinformatics analysis, but it's really interesting when you were analyzing senders and receivers, knowing that, and you were able to assess the direction. How is this being done? Like how? Yeah.
Yeah, I think the way we've been looking at that and how the package does this is to see what cells are expressing the ligand or the gene associated with the protein for a ligand, and where the gene for the receptor is being expressed. And what we do is we specify the cells of interest as well. So say if we're looking between our cancer-associated fibroblasts and epithelial cells, And then we go through and see the ligand receptor pairs between those two cell types and identifying what the major interactions as per the CellChat database are what's popping up. So that's how we go about that analysis.
Yeah, thank you. That makes absolute sense to me. So yeah, cool. Thank you. Brett?
I know we talk a lot to people about single cell data sets. They're often very helpful in designing custom panels. Can I ask, just like in terms of the value that you found of having those single cell data sets, what was your thinking on how valuable they were to being able to understand some of the biology in this case?
Yeah, it's a great question. I think when we were starting panel design, we didn't quite have the luxury of referring to other spatial data sets, or in this case with Prime, to try and see what markers are actually being expressed well utilizing Xenium. So I think we were really considering the next best thing, and the next best thing for us was the single cell data. Now, I have to say that some markers have worked well when we compare between single cell RNA sequencing as well as Xenium, but some markers don't. And so that is still something that we're trying to figure out, is how can we optimize panel design as we move further with our experiments. So they definitely are of use. And I know that when panel design is being evaluated, it's still being evaluated using single cell references. So I think that that's still, in my mind, the go-to method. And the hard part is when we want to add in genes of interest, which aren't as well reported in terms of expression in single cell or with spatial data, and we're just adding them because of their, historical significance and certain pathways, that can be much harder.
Cool. I mean, it seems that the spatial did a good job of defining those tissue regions compared to your pathologist. Was that a surprise to you?
It wasn't in particularly a surprise. I mean, we do have a lot of faith with Xenium, otherwise we wouldn't be using the technology. I think it was good to just verify it, because that is what we consider clinical ground truth to be in day-to-day clinical practice. The hard part is that the pathologist is not going to annotate our slides cell by cell. So we're not going to have that direct matching. We can only really look at general areas. But I think it was giving us some confidence, definitely.
Did you find any surprises in... So you said that there is a good correlation between what pathologist annotated and what Xenium did. I remember, if maybe you can go back to that slide, in this cancer region, I think there were like a bit few different, maybe normal epithelial, right? And it's a bit less blue than... So do you think there are some surprised findings here that Xenium actually helped with?
Yeah, potentially. I mean, it's still hard. We recognize that a lot of what we picked up as normal may, in fact, be myoepithelial cells, which are lining ducts, and that is part of normal breast architecture. So perhaps what we're identifying is a cluster that is more myoepithelial in expression. And there still is, in our mind, some difficulty just by using gene expression to differentiate between normal epithelial and invasive epithelial. So I still think that is quite hard using just gene expression. I think ultimately that's why with single cell analysis, they end up using inverse CNV to try and tell between the two, but we don't have that luxury, unfortunately, to look at CNV with spatial. So I have to say that with those small areas that you're referring to in the top right, where there are invasive epithelials being picked up, I'm not sure if I necessarily quite believe that, intermixed amongst the normal. And we are still trying to figure out better ways of annotating epithelial cells, particularly those minor cell subsets that we're talking about.
Yeah. And because we were talking about cell-cell communication, and I'm going back to these senders, receivers, did you like zoom in into have like some specific examples that you could see? Yeah, this is sender, this is receiver. I didn't see it in this deck, but I'm just curious if you have something like that.
Not quite as yet, because we're not sure if what we're seeing may be an artifact of one method. So that's why we are trying these other cell-cell communication methods to really verify. And it's also hard when we don't quite we'd ideally want to repeat that kind of analysis when we have our minor cell type annotations, because what we're really interested again is in the heterogeneity of the tumor microenvironment. And so I think that's where the real interest lies in our minor cell annotations. But without those, we haven't really proceeded with more in-depth cell-cell communication analyses.
We have very shy audience today, I
Yeah, we're
must
getting close
say.
to time, so we'd encourage anyone, if you do have any questions, to put them into the chat. We've got another minute, which we can hold on for if anyone is interested.
And in the meantime, I can ask another question. Fascinating that part about unannotated cells. Was it the single cell part or was it the Xenium part? Because then I got lost a bit.
Yeah, it's potentially a few factors. It could be the limited gene panel, for one, that may not be picking it up, but I think more likely it's a number of cells that had low transcript. And we're trying to figure out the best method of filtering these, what we consider low quality cells. And recently there was that publication from Mats Nielsen's group about some of the pipelines that they've recommended for Xenium analysis in QCing the data. And up till now, we've kind of just been what almost feels like wandering in the dark, because there hasn't really been set gold standard methods of analyzing the data. So we are going to try and adopt some of those methods and consider things like ambient RNA removal, which, you know, that's often been used in single cell. I don't know how relevant it is to spatial data, but there are different things we're looking at.
I think we might learn in a part 2 bioinformatics when it will be coming.
That's right. Hanyun will definitely speak about those things that we tried.
Yeah, I know the Mats Nielsen paper was a great one that for those who are interested, Mats Nielsen is a foundational spatial biology researcher based in Sweden, and he's been very, very influential in the field and an early adopter of Xenium technology. Great. Well, we still don't have any questions, so sign of a great talk is what I think. So look, Jeremy, we'd like to thank you for coming on today to have the conversation, and yeah, good luck with the research.
Thank you.
Yeah.
Thanks, everyone.
Thank you. Bye.
Thanks, everyone.